I am going to discuss one more important chapter, that is immunology or immune system, and we are going to discuss this as an integrated teaching, and the microbiology department is going to integrate with us. First of all, I would like to say thanks to Dr. Rajani, Professor and HOD Department of Microbiology, and also my sincere thanks to Dr. Dhanlakshmi, Associate Professor in the Department of Microbiology. Okay, today I am going to discuss chemistry of immunoglobulins. It is my part, and Dr. Dhanlakshmi is going to discuss about immune system, different types of immune system, and what do you mean by innate, adaptive immune system, immune response, immune tolerance, autoimmunity, type of vaccines, and hypersensitivity reactions. Of course, this is a very big and important topic. We are going to discuss in brief. You are going to learn in detail about this immune system in microbiology when you enter into the second year. Coming to chemistry of immunoglobulins. Immune system consists of three major components, that is B lymphocytes, T lymphocytes and innate immune system. B lymphocytes are mainly derived from bone marrow and are responsible for synthesis of circulating antibodies. They are also called as immunoglobulins. T lymphocytes are of thymic origin and are involved in variety of cell-mediated immunological process. The innate system defends against infections in a non-specific manner and are non-adaptive, containing variety of cells such as phagocytes, neutrophils, natural killer cells and others. Coming to chemistry of immunoglobulins. Immunoglobulins are antibodies synthesized from B cells. They are glycoproteins in nature, having Y-shaped structure. These immunoglobulins plays an important role in body's defense mechanism. In electrical field, these immunoglobulins appear in gamma region, hence they are called as globulins. Immunoglobulin is a functional term, whereas globulin is a physiological term. The immunoglobulins are having Y-shaped structure consisting of two identical light chains and two heavy chains held together as tetramer, that is L2H2 by intrachain and interchain disulfide bonds. Heavy chain consisting of 450 amino acids and light chain consisting of 212 amino acids. The heavy chains are linked to carbohydrate content, hence immunoglobulins are glycoproteins. L chain may be either of two types, kappa or lambda, but not both. Heavy chains may be of 5 types and are designated as alpha, gamma, delta, mu and epsilon. The type of H chain present in immunoglobulins determines the type of immunoglobulin like IgA, IgG, IgD, IgM and IgE. Both the light chains and heavy chains contains amino terminal and carboxy terminal end. This is the structure of immunoglobulin. The L chain consisting of one variable region VL and one constant region or domain, whereas most of the H chains consisting of one variable region and three constant regions CH1, CH2 and CH3. IgG and IgA have 3 domains, whereas IgM and IgE have 4 domains. Variable region of both light chain and heavy chain at amino terminal end have extremely variable amino acid sequences called as hypervariable region which forms antigen binding site, whereas constant region of H chain is responsible for various biological activities like complement activation and binding to cell surface receptors. There is an hinge region which forms a flexible link between Fab and FC region, and it is flexible nature which helps in flexibility of Fab fragment and also helps in binding of two specific antigens to the antigen binding site. The papain cuts the immunoglobulin molecule at a site towards the amino terminal part of the disulfide linkages. So, this papain splits immunoglobulin into two Fab and one FC portions, whereas pepsin, the enzyme, cleaves the molecule towards the carboxy terminal part of the disulfide bond so that one FAB square and one FC portion are produced. Depending upon the coming to different types of immunoglobulins, depending upon the heavy chain present in immunoglobulins, they are named as immunoglobulin A or IgA, which contains alpha H chain, immunoglobulin D or IgD, which contains delta heavy chain, whereas immunoglobulin E or IgE contains epsilon H chain. Immunoglobulin G or IgG contains gamma H chain, whereas IgM, immunoglobulin M, contains mu type of H chain. Coming to individual immunoglobulins in detail, IgA, it is a monomer or dimeric protein, second most important globulins constituting about 20% of total IgC. It does not cross placental barrier. IgA consisting of two heavy chains and two light chains, that is alpha 2 kappa 2 or alpha 2 lambda 2. There are two types of IgA, secretory IgA and serum IgA. Secretory IgA is present in cholesterol, saliva, tears, respiratory, intestinal and genital tract secretions. Coming to functions of IgA, IgA prevents the attack of bacteria and virus to mucous membrane and thereby prevent entry of foreign substances to the cells. And it does not fix complements. Normal serum concentration of IgA ranges from 70 to 400 milligram per deciliter. Its concentration is decreased in leukemia, nephrotic syndrome, ataxia, telangiectasia, etc. And some people may born with absence of IgA or less IgA also. And its concentration is increased in autoimmune diseases like rheumatoid arthritis, systemic lupus erythematosus, liver diseases like cirrhosis of liver, chronic hepatitis, etc. Coming to IgG, immunoglobulin G, it is a major class of immunoglobulin and constitutes about 70% of total immunoglobulins. It is a monomeric and it is consisting of gamma 2, kappa 2 or gamma 2 or lambda 2, and it is the only immunoglobulin that crosses the placenta and it is the only maternal antibody which protects fetus. It has two antigen binding sites released by beta cells. The function of IgG is it protects against bacterial and viral infections. It crosses placental barrier and provides protection to the fetus. And also it activates complement and abscesses bacteria. The normal serum IgG concentration ranges from 700 to 160 milligram per deciliter. Its concentration is decreased in Waldenstrom's macroglobulinemia, and in case of multiple myeloma, chronic hepatitis, multiple sclerosis and AIDS, the IgG concentration is increased. Coming to IgD, immunoglobulin D, it constitutes about 1% of total immunoglobulin. It is a monomer made up of delta 2, kappa 2 or delta 2, lambda 2, and this immunoglobulin D is a mobile molecule. The function of this IgD is uncertain. It may function as antigen receptors. Normally it ranges from 0.2 to 0.6, to 2 to 6 milligram per deciliter. High levels of immunoglobulin D indicates it is a case of multiple myeloma. Coming to IgE, immunoglobulin E, it is a monomeric protein consisting of epsilon 2 kappa 2 or epsilon 2 delta 2, and it is present only in traces amount in the blood. Immunoglobulin E, it binds with mast cells, basophils, and causes rupture of cell membrane. Also mediates hypersensitivity and anaphylactic reactions. It acts as anti-parasitic, especially for helminthic infections. IgE concentration is increased in ataxia, telangiectasia, and increased in parasitic infections, allergic reactions, asthma, some type of cancers, and also in autoimmune diseases. IgM, immunoglobulinium, it is the largest pentameric protein joined together by L chain consisting of mu2 kappa 2 or mu2 lambda 2, and it accounts for about 10% of total Ig. It is the first antibody produced in response to an antigen. It cannot cross the blood vessels because of its large size. Coming to functions of IgM, it fixes complement and also plays an important role against bacteria and virus. Normal concentration is about 20 milligram per deciliter. 120 milligram per deciliter, its normal concentration in the blood is about 120 milligram per deciliter. Its concentration is decreased in multiple myeloma, autoimmune diseases, and also some types of infections. And its concentration is increased in Waldenstrom's macroglobulinemia, early viral hepatitis, nephrotic syndrome, etc. Increased concentration of IgM indicates new infections. Thank you. And some of the important questions in this chemistry of immunoglobulin, they are going to ask, write the structure of immunoglobulin, how they are classified, what are the functions of immunoglobulins. Any one type of immunoglobulin, maybe IgA, IgM, IgD, anyone, maybe ask for 3 marks. Then you have to write only 3 or 4 sentences. If it is a 5 marks question, you have to write the structure of immunoglobulin, functions in detail. Right, one more important question they are going to ask in this chemistry part is Benja's proteins. It is related to this topic only, but I have already covered under plasma proteins. That means you have to write what are Benja's proteins, what is multiple myeloma, how we are going to detect, etc. Okay, now I invite Dr. Dhanlakshmi to continue.
Good morning everyone. Today we will learn the basics of immunology. Immunity is defined as the resistance exhibited by the host towards injury caused by microorganisms or their products. Means it is the resistance power or the protectiveness of the human host whenever microorganisms attack the host. In the bargain, this leads to protection against infectious diseases. Immunity is of two types, innate and acquired. Innate immunity is the first line of defense. It is due to the genetic and constitutional makeup and not affected by prior contact with microorganism or the immunization. Now let me decipher this sentence. This sentence means that immunity is present from birth. The immunity that is present from birth is called as innate immunity. Before we come in contact with any microorganism or any kind of immunization, whatever resistance power or the immunity that we have from birth is called as innate immunity. This innate immunity is brought about by various mechanisms like that of skin. See, this intact skin which covers the entire human body acts as a barrier for many microorganisms from entering. Mucosa. The mucosa of the oral cavity or the genitourinary cavity, all of them have enzymes or mechanisms which can prevent the microorganisms from entering the host. Mouth. The saliva in the mouth acts as a protective mechanism. It will have a lot of enzymes. It acts as a barrier for the microorganisms to enter the rest of the gut. Similarly, the tears in the eyes protect the conjunctiva from the microorganisms. Then the urine, the mechanism of flushing of urine sees to it that the organisms will not enter the urinary tract further up, rather they are flushed out. So because of all these innate immunity, the human host will not be attacked by microorganisms. Then adaptive immunity. This is the kind of immunity which is acquired during our lifetime. Means after birth, due to exposure to multiple microorganisms, we develop a kind of immunity or protection which is called as adaptive immunity. This adaptive immunity has certain characteristics, some of them being antigen specificity. Means adaptive immunity is able to identify or distinguish between antigens. For example, if the same antigen or the same microorganism again attacks the human host in the future, then it will be identified. The human host immune system knows that this is an antigen which had come previously and I had raised an immune response against this antigen. So it will be identified and it has a immunological memory. The memory cells get activated and the response this time, which we call it as a secondary response, will be much more quicker and much more aggressive. The host immune system is able to identify the self and non-self antigens. Means our own antigens are not identified as foreign. Only the antigens from microorganisms or the external environment are identified as foreign and an immune response is raised against them. The self antigens will never raise a immune response. This is called as self-tolerance. And when this self-tolerance fails, means the host is not able to identify his own antigen as his own, then it leads to a condition called as autoimmunity, wherein immune response is raised against our own antigens. This condition is called as autoimmunity. Now, to put it in a nutshell, our immune system, which is our ultimate line of defense, has barriers, internal barriers and external barriers. The first line of defense in the immune system are our phagocytes, natural killer cells, and second line of defense are the T cells and B cells, both of which lead to adaptive immunity. The phagocytes and natural killer cells lead to what we call as innate immunity or the immunity from birth. Now, immune response. This is the reaction of the host to any antigenic stimulus. This immune response can be humoral or cellular. Humoral means whenever there is an antigen exposure, the human host raises an immune response by production of antibodies. This is called as humoral immune response. And whenever there is an antigenic stimulus or an exposure, the host raises an immune response by increasing the number of activated cells. This is called as cellular immune response or cell-mediated immune response. Now, if you look at this picture, on both sides, on one side I have depicted antibody mediated immune response and the other side is cell mediated immune response. Whenever there is an antigen which enters into the human host, both the immune responses are activated, that is antibody as well as cell mediated. But in some infections, antibody mediated immune response is more important, and in some, cell mediated immune response is more important. The ultimate goal is to remove or kill this antigen or the organism which has this antigen. When the antibody mediated immune response is activated, the B cells, I told you the B cells are important in generating the antibody mediated immune response. B cells become converted to the plasma cells and start producing a large number of antibodies. These antibodies go and attack the antigen. They are very, very specific to the antigens. Where are the antigens present? They are present on the microorganisms, or the cells that contain the microorganism will exhibit this antigens on their surface. So the antibodies go and bind to that. They either kill the cell that contains the microorganism or the microorganism per se. That's how the antibody mediated immune response works. Then the cell mediated immune response. I told you the cell mediated immune response is brought about by the T cells. T cells are the most important cells bringing about the cell mediated immune response. What actually happens in a cell mediated immune response? Whenever there is an antigenic stimulus, the antigen is first phagocytosed by all the macrophages. The tissue macrophages or the circulating macrophages will first phagocytose the antigen. Then it is broken down into minute small pieces and the antigen of it is identified. This antigen is presented or it is transported to the surface of the T cell. So it is present on the surface of the T cell, which is identified by the T cell receptor, due to which the T cell get activated. So the antigen which is present on the macrophage's surface is identified by the T cell receptor of the T cell, which leads to activation of the T cell. This activated T cell now starts releasing large amount of cytokines. These cytokines are responsible for forming pores onto the infected cells or the microorganisms themselves, and due to the formation of pores, either the cells or the microorganisms get killed. This is how the cell mediated immune response acts. Now, this is in a nutshell. Whenever there is an infection or an infective agent or an antigenic stimulus, both the limbs of the immunity are activated, that is the humoral immune response and the cell mediated immune response. In the humoral immune response, the B cells become activated and they become large in size, called as plasma cells, start releasing large amount of antibodies. The antibodies will go and attack the antigen or the antigen containing cells, which leads to killing of the antigen. Simultaneously, the macrophages will phagocytose the antigen and they will present it to the T cell. The T cell identifies the antigen on the surface of the macrophage by its T cell receptor and it starts releasing large amount of cytokines. The cytokines lead to pore information or pores on the infected cells and they will kill the infected cells. This is how cellular immune response or cell mediated immune response is brought about. Then immunological tolerance. Immunological tolerance means unresponsiveness or contact with any antigen abolishes the capacity to mount an immune response against that particular antigen when administered subsequently. This is called as tolerance. Means whenever we expose a human host to an antigen, he is supposed to raise a immune response. He is supposed to eliminate this antigen, but in sometimes what happens is it is not identified as a foreign antigen. The host immune response will not raise any response at all. This is called as tolerance, means the immune system is tolerating this antigen. This leads to the infection becoming more serious and the immune system may not be able to tackle this infection. This happens due to, there are various theories of why immunological tolerance happens. The various theories are explained. One is elimination of the T cells. So when T cells are not there, so there may be a genetic abnormality wherein the T cells are not there, or the ones who are there, they are abnormal. So that will not identify the antigen at all. There will be no B cell activation, hence no production of antibodies. This may lead to immunological tolerance. Or there may be immunocompetent cells which will interfere with the immune response that can be raised, or there may be antibodies which are synthesized, they may be abnormal, or they may be neutralized by the host himself. In all these conditions, the host immune system will not raise any kind of response. That is called as immunological tolerance. Now, autoimmunity. I told you autoimmunity is a condition wherein immune response is raised even to self antigens. That is autoimmunity, and various mechanisms are explained. Antigenic alteration. See, antigenic alteration is something that happens in microorganisms very often. The organisms change their antigen and host immune system is not able to identify this new antigen. It will show it as a new antigen. It will perceive it as a new antigen. So again it has to go through production of antibodies or the mechanism of identification. So in that, autoimmunity may be raised. And sequestered antigen. Sequestered means during the embryonic life, these antigens are not shown to the host immune system. The best example is the protein of the human lens in the eye. The lens protein is never exhibited to the host immune system during the embryonic life. So in the future life, whenever the lens protein gets released into the circulation, see, the cause may be a traumatic rupture of the lens, or during the cataract surgery the lens protein which gets spilled over into the circulation, it will be identified as non-self or a foreign antigen. That time autoimmunity may be seen. Then molecular mimicry. Molecular mimicry means some microorganisms have antigens which are similar to proteins which are present in human host. So the antibodies and the activated T cells which are produced against the microorganism will start acting on host antigens, that is self antigens, which will lead to again autoimmunity. Sometimes there may be polyclonal B cell activation, means large amount of B cell activation occurs, which leads to production of large amount of antibodies which are non-specific and they start attacking the self antigens. For that also autoimmunity may be seen. Forbidden clones. Forbidden clones means in the embryonic life, again whenever an antigen is identified and an immune response is raised against it, the host immune system is forbidden. It is not seen such clones, and suddenly when such clones are seen during the lifetime, it leads to formation of large amount of immune response or antibodies, which is called as autoimmunity. Some of the examples of autoimmune diseases are Hashimoto's thyroiditis, Graves' disease which is thyrotoxicosis, myasthenia gravis, pernicious anemia, SLE or systemic lupus erythematosus, and rheumatoid arthritis. Then coming to artificial active immunity. This immunity, initially when I told you there is innate and acquired immunity. See, acquired immunity is acquired during our lifetime, either due to exposure to the organisms or due to vaccines. This immunity that we get through vaccines is called as artificial active immunity. Active because it activates the host immune system to produce immune response, and artificial because we are giving it artificially. The person is not exposed to the microorganism. He is not getting infection during his lifetime. He gets something similar to the microorganism through the vaccines, in the form of vaccines, because of which the host immune system gets activated and it produces the immune response. That's why it is called as artificial active immunity. There are various types of vaccines like live, killed, subunit vaccines. Live vaccines means they contain very low or less virulent or less dangerous but living microorganisms. The microorganism is viable, means it is alive, but it is not as dangerous to cause a disease. Examples of live vaccines are BCG for tuberculosis or oral polio vaccine. Then we may have vaccines prepared from killed preparations of the same microorganisms. Means the microorganisms are grown, they are cultured, then they are killed. The killed filtrate can be used as vaccine. Examples of killed vaccines are cholera vaccine and injectable polio vaccine. Then the other type of vaccine is subunit vaccine, wherein we don't require the whole microorganism either in the live form or the killed form. Only the antigenic component is derived and it is given to people or the public as vaccine. Examples of subunit vaccine are typhoid VI vaccine and hepatitis B vaccine. In hepatitis B, the surface antigen is separated or purified and given as vaccine. Means here we do not give the virus either in the live or the killed form. It is only the antigen which can cause or which can raise immune response in the human host is used as vaccine. They are called as subunit vaccines. Similarly, some bacterial products can be used as vaccines. Now there are certain bacteria which can release toxins, and these toxins are responsible for the diseases that they cause. So this toxin is inactivated by a procedure called as toxoiding, and the inactivated toxin is called as a toxoid. This toxoid itself can be used as vaccine. So for example, the tetanus toxoid is a toxoid. The diphtheria toxin can be toxoided and used. It is used in the form of a trivalent vaccine called as DPT, wherein it is preventable to diphtheria, pertussis and tetanus, wherein the diphtheria and tetanus are toxoids and the pertussis is a whole cell vaccine. So it is a trivalent vaccine. Recombinant DNA technology is used for production of this subunit vaccines. Recombinant DNA, which uses the DNA mechanism of the organism, means only the gene responsible for the production of that particular antigen is used. That gene is incorporated into a vector. The vector may be a fungus like a yeast, or it may be other bacterial cell, or it may be a bigger virus. So when it is incorporated into these other organisms, because it is a gene coding for the production of that antigen, large amount of that antigen can be produced. So that antigen as a subunit vaccine can be used to give protection. That is recombinant DNA technology. Now a word on hypersensitivity. Hypersensitivity is rather opposite of immunity, means it is overexpression or it is injurious, undesirable injurious consequences in the host following contact with specific antigens, which this hypersensitivity we also call in layman language as allergy. Means the same antigen which does not cause a disease in others may cause in few. This is allergy. You all are aware of allergy, that is due to the hypersensitivity reaction. This hypersensitivity may be immediate or delayed based on the time required by the host to develop a clinical reaction, or this over enthusiastic injurious reaction. Immediate hypersensitivity is B cell mediated and delayed hypersensitivity is T cell mediated. Thank you.